The mitochondria, which is the powerhouse of the cell, contains its own mitochondrial DNA, which is maternally inherited and responsible for the synthesis of electron transport chain complexes. These complexes are responsible for the mitochondria's job of oxidizing ingested fats, proteins, and carbohydrates, and using oxygen to replenish ATP for the cell. As humans migrated out of Africa and into different environments, the mitochondrial DNA picked up mutations in response to different environments. These mutations were energetically beneficial to the environments. For example, in the warmer climate of Africa, the mitochondrial DNA remained relatively unmutated, as a result, generating more ATP and consuming less oxygen, and also producing less excess body heat. Whereas in the colder environments of Northern Europe, the mitochondrial DNA in these populations picked up mutations that allowed for greater generation of excess body heat, but a decreased efficiency of oxidative phosphorylation, thus generating less ATP and consuming more oxygen. Epidemiological studies have determined that these mitochondrial DNA variants, which we call haplogroups between these different regions, are key modulators for sensitivity to both chronic and transmissible disease. And there remain relatively few details known about functional differences between the African and the European haplogroups. In this study, we aim to determine if there are such differences, cellular differences between the African and European haplogroups. To do this, we collected lymphoblast samples from patients from the African haplogroup L4 and the European haplogroups H, J, and UK. We took these lymphoblasts and we fused them with mitochondrial DNA deficient A549 lung carcinoma epithelial cells, generating independent transmitochondrial cell lines, which we call cybrids. And these cybrids differed in their mitochondrial DNA background, but contained the same nuclear background, thus providing us with an effective model for studying cellular differences that come from mitochondrial DNA backgrounds. To determine if there were differences in transcriptional profiles, we cultured the cybrids at basal conditions and in parallel, and collected RNA samples and performed RNA sequencing analysis as well as pathway analysis from our RNA sequencing. We found that over 15% of the total transcriptome is differentially expressed between the African and the European cybrids at a p-value of 5%. Using pathway analysis, we found that the pathways with greatest enrichment in the African cybrids relative to the European cybrids at baseline conditions were involved in amino acid metabolism, the integrated stress response, and antioxidant defense. We also aimed to determine energetic differences between our African and European cybrids and performed a mitostress test using the Seahorse XF Pro assay. In the mitostress test, we found that our African cybrids had lower basal and glycolytic extracellular acidification rate compared to the European cybrids, but a higher non-mitochondrial oxygen consumption rate compared to the European cybrids. This study shows that subtle mitochondrial DNA differences between the African and the European haplogroups can induce these large transcriptional and energetic differences in our cybrids, and that these cybrids could serve as a useful tool to continue studying differences in haplogroups, which may ultimately provide us with greater insight into cellular mechanisms underlying differences in sensitivity to disease among haplogroups.